People have said that the eyes are windows into the soul. Well, that's poetic, but how do these windows allow us to perceive the world around us? Hi, I'm Rich Burnett for Wondrium, and in this episode of Perspectives, 5 experts delve into the world of vision, from how we experience sight to the biology of how we interpret those signals in our brain. Let's begin now with a crash course on the mechanics of how our eyes actually work.
How much do the mechanics of vision play a part in our perception, and therefore our experience of the world? Well, quite a bit. To understand it, we need to start with the physiology of seeing. Let's assume we are talking about a healthy person with 2 eyes, and average vision. First, the ability to see involves a 2-step process. We experience sensations in our eyes that are translated to perceptions in our brain. Sensations, perceptions. 2 different things that both contribute to the phenomenon we think of as seeing. Let's start with the sensation part, the part that happens in your eyes. Everything we see starts with light. In fact, the only thing we ever see from a sensation standpoint is light, and our eyes are exquisitely built to gather it and respond to it. The cornea, iris, lens, and retina make up the anatomy of our eyes. The retina contains light-sensitive tissue. Light passes through the cornea and is bent or refracted through the pupil in the iris and onto the lens. The lens in your eye is responsible for taking the refracted light that passes through your cornea and adjusting its refraction so that the image is in focus when it hits your retina. The cornea itself already does a lot of the focusing as it bends the light to pass through the iris. But the curvature of the cornea is relatively static. It doesn't change based on the distance of what you're viewing. To accommodate different focal lengths, we need the lens. The lens is surrounded by muscles which contract or relax based on the distance of your view. If you're looking at something far away, these muscles are relaxed, allowing the lens to flatten and bend the light less. If you're looking at something close up, the muscles contract, rounding the lens and increasing the refraction. All of this is happening in less than the blink of an eye, with no conscious effort from you, until you reach your 40s at least.
The retina is the location of photoreceptor cells which contain proteins called opsins that detect light. Opsins are transmembrane proteins coupled to G proteins. The distinctive feature of opsins is that they hold a molecule of vitamin A that gets altered when exposed to light. Thanks to opsins, photoreceptor cells detect light and pass signals on to other neurons called ganglion cells. The axons of the ganglion cells are bundled into the optic nerve, which routes the information, ultimately, back to the visual cortex in the brain to process the signals and tell us what we are seeing. There are 2 types of retinal photoreceptor cells involved in light detection: rods and cones. Rods are responsible mostly for detecting light under the very low light conditions. They provide little color information since they are optimized to detect things in very dim light. What rod cells lose in color detection, they gain in sensitivity. A rod cell can detect a single photon of light. Color detection is the domain of the cone cells. Humans have 3 types of cone cells, each specialized to absorb wavelengths corresponding to red, green, or blue light. Cone cells operate best in bright light.
The workhorse of both the rod and the cone cells is a molecule called retinal. Your body makes retinal first from beta-carotene, a pigment that gives carrots orange color. Your body then converts that beta-carotene into vitamin A and subsequently into a molecule called retinal. What's special about retinal is that it has a carbon chain that can take 2 different configurations, cis-retinal and trans-retinal. Chemists call that 2 different isomers. They're different physical shapes of the same chemical formula. In its usual configuration in the eye, retinal takes what's called the cis conformation. You see how this is a regular zigzag carbon chain for carbons at positions 7, 8, 9, 10, and 11. But at carbon number 12, it takes a bend. That bend is why this is called the cis conformation. Cis comes from a Latin root meaning on the same side. Off of each carbon in the chain is a side group. Those are things like the hydrogen atoms. When two side groups in a row are on the same side of the chain, that's called a cis configuration. Here you can see where the hydrogens associated with carbon atoms number 11 and number 12 are on the same side of the chain. So at that bond we have a cis configuration. The other isomer of retinal is called transretinal. In this configuration, the zigzag no longer takes a bend. It's been straightened out into its transretinal conformation. It's called trans because now the hydrogens are on opposite sides of the double bond between carbon atoms number 11 and number 12. In fact, the longer name is all transretinal, since the side groups alternate sides along the entire chain. The key to vision is that absorption of a photon in the right energy range converts the retinal molecule from its usual bent configuration, which is the cis form, into the straightened out trans configuration.
This shape change for retinal also changes the rhodopsin holding it, resulting in activation of an intracellular G protein called transducin. The next few steps involve sodium ion channels. In the absence of a signal, these channels are kept open to allow sodium ions to move across the membrane. When activated as a result of light detection, transducin causes an enzyme called cGMP phosphodiesterase to catalyze breakdown of the molecule propping open the gated sodium ion channels and calcium ion channels. This keep the channel open molecule is called cGMP or cyclic GMP, and it's normally present in the cone and rod cells in the absence of a light signal. In other nerve signaling you have seen, it's the opening of the sodium channels that initiates the nerve signal. Here, it's the closing of the channels that starts the signal. Anyway, each detection of light by a photoreceptor leads to cyclic GMP within it being broken down by the activated cGMP phosphodiesterase, and the gates to the sodium and calcium channels close. This has the opposite effect from what you have seen before. Instead of an opening of ion gates that leads to depolarization, what we have is the closing of gates. Sodium and calcium are not able to enter, and we have hyperpolarization of the membrane. This hyperpolarization triggers downstream signaling that ultimately relays information to the brain, which assembles this information from all the photoreceptors and paints the pictures for us that amount to vision.
The information that streams from the retina through the optic nerve requires massive decoding in order to generate visual consciousness. That decoding takes place through a number of steps at a number of different places in the brain. An optic nerve leaves each eye. They meet at the optic chiasma, and there the information is sorted into two batches. Information from the left side of each retina, the left side of each visual field, is routed to the right hemisphere of the brain. Information from the right side of each retina, the right side of each visual field, is routed to the left hemisphere of the brain. On each side of the brain, that divided information from the retinas is transferred via the lateral geniculate nucleus, a way station in the thalamus, to the back of the brain. Put your hand over the back of your head, and you've put it over the occipital lobe of the brain. That's the visual cortex, and that's where the decoding of the message from your eyes really begins. Even there, processing doesn't happen all at once. Different areas of the visual cortex have been found to process different aspects of the stream of information from the retinas with quite different effects. The first area of information arrival is known as V1, Visual Processing Area 1. That's the area of your brain that comes closest to an inner screen. A map of retinal impulses is laid out over V1, though the map's a bit distorted. It tends to map things out in ways that correspond to the intensity of information coming in from the eyes. Since information is most intense in the fovea, or the area of focus, a greater area of V1 is devoted to that area. The representation that appears on V1 is laid out like what you see in a fisheye mirror. V1 is just the first stop. In the adjoining area V2, stereo vision is processed, putting together the information from two eyes to generate your impression of a three-dimensional scene. V3 is a distinct area that processes depth and distance. V4 is devoted to color, V5 to motion, and V6 is at work processing the position of objects. One of the things that you see is where things are. Another thing you see is what they are. Researchers often refer to a where path, identifying spatial location, as starting in V1 and proceeding through V2, V3, V5, and V6. The what path goes from V1 to V2 and V4. But it doesn't stop there. Processing continues further into the brain, each area having a specific role. There are at least 30 distinct areas in the brain devoted to different aspects of visual processing. In a very real sense, then, the visual scene of which you are conscious doesn't come from the eyes. It's the result of a complex of multiple bits of decoded information assembled from very different areas of the brain.
Impossible figures and drawings illustrate everyday challenges we face when trying to move from sensation to perception. It's not as simple as just perceiving what we see or hear or feel in the world. Sometimes this sensory input can be perceived or interpreted in different ways. Early in the 20th century, so-called Gestalt psychologists were among the first to study how the mind organizes sensations into a perceptual gestalt, the German word meaning form or shape. They recognized that we automatically organize isolated objects we see in the world to form more cohesive wholes. The most fundamental Gestalt principle of organization is our ability to distinguish an object from its surroundings, the figure from the ground. Black and white images are often used to illustrate surprising switches between what is perceived as foreground and what is perceived as background. One classic illustration of the figure-ground problem can be perceived as two black silhouettes of people facing each other with a white background in between them. But this same image can alternatively be seen as a white vase, with the black on either side appearing as the background. Images that allow for more than one possibility show how our perception organizes what we see. A more difficult version of this perceptual switching occurs in images cleverly constructed to allow you to perceive two different figures within the same region, while the background region remains unchanged. One famous example portrays one image of a young woman, neck exposed, looking away, while the other is of an old woman in profile with a larger face and a much larger nose and chin. In both of these examples, the brain is sorting out how to define the figure, the thing to focus on, and how to define the ground, the things to ignore.
Okay, on to the other Gestalt principles, which are a little more straightforward. The second Gestalt principle that operates during perception is closure. The idea of closure is simply that our brains want to close open-ended forms or fill in gaps if there are pieces missing. The next principle, continuance, is the idea that we will always follow visual lines, curves, or sequences as we try to make perceptive sense of something. This principle is so strong that our eyes will follow a path, a river, a fence, or a horizon line right off the map, so to speak, literally out of the frame of an image. The fourth principle, the law of common fate, suggests that your brain visually perceives two or more things moving in the same direction as basically one unit. Think of a group of geese traveling in V formation across an afternoon sky. Your brain doesn't perceive that image as 5 or 7 or 9 different geese. It perceives that image as a single flock of geese, a V of geese even, one thing. The principle of similarity is, well, similar to common fate. But instead of our brains using motion and direction to establish unity, they use similarities in forms, colors, patterns, or other attributes. Our brains do not see 150 individual red apples sitting in the grocery store. They see a pile of apples, all red, all roughly the same shape. We don't really see two identical Argyle socks, we see a pair of socks. Finally, the last Gestalt principle of perception, proximity. Proximity is simply the idea that things that are close to one another are part of one group. This has nothing to do with direction or form or color or anything else. You can take any random collection of items, put them in close proximity to one another, but separate from anything else, like in a stack or a pile, and your brain won't look at it and say, hey, look at that paperclip, banana, fedora, magazine, and shoelace. No, the law of proximity says that your brain will automatically try to organize that image as one thing. Hey, look at that thing, that piley thing. What is that thing?
One fundamental question is whether our ability to organize sensory information is innate, or whether this ability develops over time with experience in the world. People who are blind from birth but later undergo surgery and gain the ability to see suggests that some visual abilities, such as the ability to see color, result from fundamental biological processes. But other abilities, such as recognizing objects, seem to require experience in the world. Studies using animals have shown that without early exposure to normal visual experiences, such as when an animal's eyelids are sewn shut from birth, certain parts of the brain fail to develop even when their ability to see is restored. This finding suggests that early visual experiences during the first few months of life, the critical period, help guide the development of neural connections in the brain. So, at least for vision, it appears you need to use it or lose it, or at least use it soon or lose it. Organizing sensory information from the world into coherent and meaningful units is obviously an important part of perception. But perhaps the most fundamentally important part of perception is the final stage, how we interpret that information. Your perceptual abilities would enable you to recognize the shape of a person standing outside your door one afternoon, but it's the interpretation of that person's intent. Are they selling Girl Scout cookies, soliciting a donation, promoting their religious faith, that would influence whether you'd open the door?
Thanks for watching. If you'd like to learn more about the topics in this episode, the full list of series that these clips came from is in the description below. You can watch them all now on Wondrium.